Okay, well, hello everybody. I hope by now everybody's been able to join this webinar. A very warm welcome to all of you. My name is Julia Buckingham and I'm the chair of the NC3Rs, and it's my huge pleasure to welcome you to what I think is a very special event to celebrate the award of the NC3Rs prize. Now, before I go any further, I need to tell you that this session is being recorded, and that's so that we can put a video of the session on our website afterwards, and people who couldn't join us this afternoon will have the opportunity to see it at a later time. Now, this celebration is, I think, particularly special this year because it marks the 20th anniversary of the NC3Rs prize. And from the outset, this prize has been sponsored by GSK. And so I would like to say a very, very big thank you to GSK for their huge generosity over the years. And I'm really delighted that this afternoon we're joined by Andy Nichols from GSK, who will say a few words before we hear from our prize winners. Now, the prize winner, as I'm sure you know, is Dr. Peter McPherson and his team from McGill University in Canada. And after we've heard from Peter, we will hear from the highly commended paper, the runner-up, Dr. Renata Nundes, from the Oswaldo Cruz Foundation in Brazil. Now, as I'm sure you all know, the prize recognizes outstanding published original scientific research that has or has the potential to have a major impact on the replacement, the reduction, and the refinement of the use of animals in research. And that, of course, is what the NC3Rs has absolutely at its heart. This prize recognizes a piece of work that's been published in a peer-reviewed journal. And very importantly, it's open to researchers from right across the world in both academia and industry. This year, we received 26 applications from 11 different countries, and that's pretty good. Now, the panel that reviewed the applications and made the decisions was also international in nature, and it had members from the UK, Europe, America, and Canada. So a very big thank you to them, because it is a lot of work to review all those papers, and we're hugely grateful for them. And the way in which they go about scoring the papers is, firstly, we score everything against very, very clear criteria. So we end up with a list of scores from which we can create a shortlist. We then send the shortlisted applications back out to the panel again for another detailed review, and then the panel comes together and discusses those papers. And that panel meeting is quite challenging. I certainly find it quite challenging to chair because we have so many excellent papers, and of course we have to come to a decision. Now, the criteria against which we measure the papers are, firstly, high quality science. And if something isn't high quality original science, then it really fails at the first hurdle. No matter how great its 3Rs potential is, it's got to be good science, and that's absolutely vital. After the science, we come to the actual or potential for a 3Rs impact. And that is very, very important too. So once you're over the first hurdle, the next really important hurdle is the 3Rs. Then we look at what the applicants have done to disseminate the information, and particularly what they've done to disseminate it beyond their peer-reviewed publication. Because, of course, what we really want is not just a really lovely paper sitting in the literature which no one looks at. What we want is a paper that people are reading and they're using that research and they are changing the way in which they work. And that's the way, of course, that we get impact. So we're very, very interested in what the team has done to try and get that impact. And then, of course, we'd like to know what you're planning to do with the prize money. And we want to make sure that you're using it wisely, as I'm sure you would do. So that's the background to it. Now it's time to move on with the ceremony itself. And it's my pleasure to introduce Andy Nichols, who's the Executive Director and Global Bioimaging Head at GSK. And he's going to say just a few words.
Thank you very much, Julia. Well, good morning, afternoon, evening, wherever you're watching this in the world. Thank you for joining us today. It is an honor and a privilege to be participating in this event and representing GSK in our partnership with the NC3Rs, which, as Julia said, has been running for the last 2 decades. This particular award, recognizing that the improvements in replacement, refinement, reduction in the use of animals for scientific research, is a huge opportunity annually to recognize both the advances in science and the improvements in ethics that we see in this particular area, particularly in my space for the supporting the development of biomedical discovery. For that 20 years of partnership with the NC3Rs, we've underlined the importance of the 3Rs in our research, a commitment that is integral to who we are at GSK and enshrined within our policies and practices within the organization on a daily basis. For these last 2 decades, we have been steadfast in our commitment and partnership. We've supported through financial investments such as this particular prize, in-kind expertise, and most importantly, a shared vision for the continued improvement in research. As we look at the progress we've made together, it becomes clear that through our collective efforts, we've not only driven scientific excellence, but continue to raise the bar for animal welfare globally, an excellent result. Science is at its core a journey of discovery and responsibility. At GSK, our ambition is to prevent as well as treat disease with well-differentiated medicines and vaccines in our core therapeutic areas. An ambition that aims to positively benefit the lives of 2.5 billion people by the end of this decade. And to do that, we apply a substantial amount of scientific research, and through that we achieve that ambition by continuing to unite science, technology, and talent to get ahead of disease. Thanks to technological breakthroughs, including the application of increasingly diverse complex in vitro models, sophisticated computational approaches, in silico models, we continue to advance how we can answer many of those complex scientific questions using non-animal methods. Such advances speak to the spirit of innovation, creativity, and scientific rigor that this award recognizes. We also recognize that there remain scientific questions for which animal studies are still necessary. Where alternatives are not yet available, we are absolutely committed to the highest standards of responsibility. The 3Rs underpin every aspect of our research, from robust experimental design and independent scientific peer review, timely application of advances in new technologies to support improved animal welfare, the maximization of the data we obtain from those studies, and the continued improvement of our welfare practices based on advances in current animal welfare science. This award and our awardees today demonstrate that through continued advancement in science and technology, whether through the wider use of in vitro derived recombinant antibodies or in vitro tox testing to support antivenom development, we continue to advance our knowledge through the applications of alternative approaches. They inspire us with their imagination, their diligence, and their unwavering commitment to the 3Rs. As we recognize the achievements today, we can reflect that our continued progress depends on our shared commitment. By both supporting and recognizing excellence in the replacement, reduction, and refinement of animals in science and research, we continue to simultaneously advance groundbreaking science itself and animal welfare. Finally, I'd just like to say that, as Julia has indicated, selecting winners is never an easy task, given the breadth and caliber of the applicants. I want to extend my heartfelt thanks to all those who put forward their work, to the review panel who undertook that diligent task and difficult task, as Julia just described, and of course, many congratulations to our 2 awardees today who are being recognized in this ceremony this afternoon. Your achievements provide inspiration for us all. So Julia, I'd like to hand back to you.
Well, thank you very much indeed, Andy, for that lovely introduction. Okay, well, let's move on now to hear from our winners. And first of all, we're going to hear from the winner of the 3Rs prize. And this, as I'm sure you all know, was awarded to Dr. Carl Laflamme, Dr. Riham Ayoubi, and Dr. Peter McPherson from McGill University in Montreal. And their paper was entitled Scaling of an Antibody Validation Procedure Enables Quantification of Antibody Performance in Major Research Applications. And it was published in February last year in a journal called eLife. Now, this paper applied a systematic and scalable characterization approach to a panel of over 600 antibodies, and found that non-animal-derived recombinant antibodies perform better than traditional animal-derived monoclonal or polyclonal antibodies. This paper, from members of the Y-Charis initiative, presents a strategy to screen new high-quality alternatives to animal-derived antibodies, building confidence in their use and driving uptake among the research community. The prize panel acknowledged that poor quality antibodies are a major factor in scientific reproducibility and commended the robust approach the team have taken to the validation of antibodies, providing convincing evidence that recombinant antibodies are not only of high quality, but often outperform animal-derived antibodies. The panel also applauded the established partnerships with the private sector, which has resulted in 73 inadequately performing antibodies being withdrawn from the market, and suppliers updating their recommendations for the use of 153 antibodies. The 3R's impact of this work, both current and future, were considered to be significant. By highlighting the substantial benefits of non-animal derived reagents can offer and building confidence in their use, this will drive up amongst the research community to advance the reduction of animal use in the development and the production of antibodies. So Peter, over to you. We're looking forward very much to hearing your presentation.
So what a wonderful way to start the day here in Canada at McGill University. And thank you so much to the NC3Rs and to GSK, Julia and Andy for this, recognizing this, our work and acknowledging our work. And as Julia said, this work is really the work of Carl Laflamme. He's the guy that's been driving this thing. Fortunately, he's giving a job talk today. So he's actually on the market to be an assistant professor. And that's I'm sort of taking his place. But the work has been done in my laboratory at McGill University. I'm the senior scientist on the project. So we're just absolutely delighted to be part of this ceremony and to receive this award. So antibodies. If you know, as just about everybody on the planet, you were exposed to the SARS-CoV-2 virus, or you were immunized for the SARS-CoV-2 antivirus, your immune system generated antibodies, and those antibodies recognized the SARS-CoV-2 virus. The immune system generates antibodies, you know, as a natural mechanism to defend against pathogens and other forms of sort of infection that might infect a human. And so basically what happens is you're exposed to a foreign antigen. That foreign antigen in something like a virus leads to the production of an antibody. And an antibody is really just a protein-protein interactor. It's a protein binder. So that antibody binds to some particular protein on the foreign pathogen and helps eliminate that protein. And of course, then in science, the antibody becomes a very important tool. It becomes a tool in that you can use you can immunize an animal. That's traditionally how these things were done. The animal recognizes the protein of interest, and now you can generate a really high affinity protein, a really high affinity antibody that can recognize the protein of interest and help the scientific study. So antibodies are, of course, amongst the most used research tools. Now, very few people make antibodies anymore in their own laboratories. We tend to purchase antibodies from commercial manufacturers. And some percentage of commercial antibodies do not perform as advertised. But what that means has been mostly anecdotal. And one of the things of our study is we've tried to quantify that. This has been a problem for years. So there's really no standardized criteria or SOPs for how to make antibodies, how to validate them. There's no comprehensive or independent framework to validate across different applications. There's batch variations, there's variable validation methods. And of course, there's two issues here in this that are really important for animals. One is that antibody production is a major use of animals in research. And the second is that the use of bad antibodies leads to flawed science and thus unnecessary use of animals. Okay, so if you're using animals to make antibodies, and half of those antibodies aren't working, then you're really doing not only bad science, but you're really doing a harm to animals. And what we kind of realize is that antibody characterization or antibody validation, or the ability to really figure out good antibodies, it's not really a science problem anymore. It's a business problem. So it's expensive to test antibodies. You know, it costs maybe $10,000 to $20,000 per antibody. It's not really cost effective when an antibody sells for maybe $500 and some high percentage of antibodies don't sell. Scientists say that the manufacturers, the people making the antibodies, should test the antibodies. The granting agencies in the journals, they want testing, but they don't have any specific requirements. There's nowhere in any of the journals or grants that say you must do this, although that we think is changing. Manufacturers believe at least some of the onus is on the scientists. Scientists say we don't have the money, testing's not required. So it's sort of this back and forth between the manufacturers and the scientists. And it's estimated that this leads to about $1 billion per annum wasted on bad antibodies. So we came up with a solution to this, and we created a new concept for antibody characterization that we call Wykeros. So Wykeros is antibody characterization through open science. And by the way, we go back and forth between Icarus and Wykeros, and we've been doing this now for 5 years. We haven't quite settled on the name. We envision Wykeros as similar to a bond rating agency, something like Standard & Poor's or Moody's, or a product rating agency like Consumer Reports, or a product safety organization. Wykeros is an incorporated company. It currently has one part-time employee who's the CEO, but all of the work is being done in my laboratory at the Montreal Neurological Institute. And the company is owned entirely by a charity. It's a non-for-profit company. So as a financial disclosure, there is no financial disclosure. There's no money being made by anyone related to Wykeros in my lab or otherwise. So, and all credit here is to Al Edwards, who is the CEO of the Structural Genomics Consortium. It was really his concept of Wykeros. And so Al came to me, had seen some work we'd done on antibody validation, and said he had this sort of concept of a company or of an organization, Wykeros, that would test antibodies. So this is the Wykeros ecosystem. And I want to spend just a little bit of time on this slide because I think it really sums up what it is we're trying to do. It's a very big slide, but it's a slide that we've been working on now for 5 years, and it continues to grow and it continues to expand. So the idea is that there is a cooperation between granting agencies, the funders, the people who want good antibodies to be used, and the manufacturing partners that actually manufacture the antibodies. And in the middle here is Wykeros. Okay. So what we do is we, over time now, as we approach, and these are the people who have funded us. So, for example, the Michael J. Fox Foundation, the ALS societies were early, came in early. In Canada, Genome Canada and Genome Quebec, the National Institutes of Health, for example. And mostly you'll notice that a lot of these funders have a lot to do with neurological disease. So Michael J. Fox with Parkinson's, ALS Society. This National Institutes of Health was very much involved in Alzheimer's disease-related research. And what the funders do is they come to us with target lists. So they say, okay, we want antibodies against Parkinson's disease proteins. So then what we've done over the years is we've built these relationships with our manufacturing partners, our industry partners. And these industry partners that you see listed here represent about 95% of all antibody manufacturing capability in the world. So we've really brought them all together. And this took a lot of time to convince these folks to join us. But now they've joined us and they're really strong partners. So we say Michael J. Fox has a list of 10 Parkinson's disease proteins. So they provide us with funding. We send that list to our partners. Our partners send us all the antibodies. And Wykeros in the middle tests all those antibodies in side by side comparisons using really well-established scientific methods. And that's why I say it's not really a business problem, a science problem, it's a business problem. Science is pretty straightforward. There's well-established mechanisms, and we've really formalized those mechanisms. So far, so Julia had mentioned a number that we'd done by the time the paper was published. Now we're up to about 1,500 antibodies tested against over 130 proteins. And so every time I present this, we update this slide. And also this idea that we need to promote this, and this is just a few examples where, we published a paper in Nature Protocols talking about the platform, the consensus platform. We've written opinion pieces in Nature for this. We had an antibody symposium in Boston where we're really trying to promote this because we really want people to understand the importance of this. And this is where I think now we finally get to the actual paper and why this is important in how it's sort of changed our understanding of antibody usage and the antibody landscape. So now that we've gone through and done, 122 protein targets and tested over 1,500 antibodies, now you're able to put science to anecdote. So the anecdote that if you ask any scientist, oh yes, antibodies don't work, the commercial suppliers, sometimes they do, sometimes they don't, but it's always anecdotal. So now the question is, what's the data say? So we do all of our antibody validation in using three approaches: Western blot, IP, and IF. So Western blot, immunoprecipitation, and immunofluorescence. And when we actually start to validate the numbers or characterize the numbers or put quantity to this, what we find is that for Western blot, and this was early on, so this was with the eLife paper, and now I say these numbers are doubled, but they're sort of holding up. So 578 antibodies that we tested were recommended for Western blot, and about 79% of them passed our validation efforts, but 20% did not. Okay, well, that's not too bad. When we came to immunoprecipitation, it got a little bit worse. So about 143 antibodies that we tested were recommended for immunoprecipitation by the manufacturers, but just over half actually were successful in immunoprecipitation using acknowledged and validated approaches. And when it came to immunofluorescence, which is probably the most difficult application, as many as 61% of the antibodies that were recommended for immunofluorescence actually did not work. But also we can go back here and look and see that there were lots of antibodies that were not recommended for immunoprecipitation, for example, that actually did work for immunoprecipitation. So we're helping the manufacturers and the manufacturers are helping us. And here's just an example of how Keros and its partners are changing the landscape. So this is an example of something called a Western blot. Got a wild type cell line and a knockout cell line, and this is for a gene called Parkin. So this is a very important Parkinson's disease gene. And so what we did was we took the way we do it, the way Keros works, is that we make a knockout cell line and we compare it to a wild type cell line. So the wild type cell and the knockout cell are exactly the same, except the knockout cell is missing only Parkin. Other than that, the genome is exactly the same. And we received from all of our manufacturing partners, 1, 2, 3, 4, 5, 6, 7, 8 times 2 is 16 antibodies. And we test them comparing the wild type cell to the knockout cell. And you can see this is a band here that's the right size to be Parkin, and it's gone in the knockout. Here's a band that looks about the right size to be Parkin, but it's not gone in the knockout. So this antibody actually doesn't recognize Parkin. This antibody from one of our manufacturing partners recognized Parkin, as did this one. And these two antibodies turn out to be the same antibody sold by two different manufacturers. And these are the antibodies that 90% of the scientific community use these two antibodies. Now, you could see they're not really that good. You don't have to be an expert to realize there's a lot of background and a lot of noise. So we took this data, we presented it back to our manufacturing partners, and one of our manufacturing partners turned around and made a new recombinant antibody. And this is the important point here for the animals. We made a new recombinant antibody against Parkin, and that's this antibody here. And now eLife, the Michael J. Fox Foundation, for example, who was very supportive of this work, is really strongly recommending to their grantees that they use this. And in fact, as indicated, our work has changed the landscape in that the manufacturers are not only either, based on our data, they're either removing antibodies from their catalogs or they're changing their recommendations. And importantly, so the first way we're helping in terms of the goals of the NC3Rs is that we're limiting the use of bad antibodies. So that's going to really eliminate the use of the need for animals in antibody production. But secondly, is that we're now really focusing on recombinant antibodies. So recombinant antibodies are almost all of the companies now have moved to recombinants. It's almost, you know, almost nobody makes a standard polyclonal anymore. So they're recombinant antibodies. They do start right now still with the immunization, but I'll be happy in the question session to talk about new approaches in which we can eliminate animals altogether from the antibody production. And that's, I think, something that's just right around the corner. But right now, so we still start with immunization of a rabbit or a mouse, but then there is a sort of a selection process where you screen out certain B cell clones, certain populations of immune cells that express the antibody. And then through PCR amplification, you're able to identify the sequences of the antibody. But then what you do is you clone those sequences into a plasmid. And now you make everything in a recombinant system in a 293 cell, in a human cell line. And you never see an animal again. And now you've got plasmids that make these antibodies in cell lines, and they're stable as could be. They'll last forever. They'll never change over time, and there'll never be a need for another animal in terms of producing that antibody. And so do those antibodies actually work? Yeah, they work. And not only do they work better. They're actually better. So we've tested them in Western blot, in IP, and immunofluorescence. And when we compare recombinants to standard monoclonals, even or polyclonals, we find that the recombinants just work much, much better. So two ways I think we're helping that the goals of the NC3Rs is to just eliminate the use of bad antibodies, and then also to really sort of reinforce and push the idea that recombinant antibodies are actually a much better way to go, and recombinants just have much, much less impact on animal use. So that's the talk. I really want to thank the people that were involved here, and in particular, I'll take the last second just to really highlight Carl Laflamme. It was his idea to apply for this. Carl has been driving this. As I say, he's off for a job talk today, so he left me to do it. And the team here, and so Al Edwards is here, and we're really, you know, one of the goals is to, I think, as Julia said, it's one thing to publish this and to develop this, but it means nothing if people don't use it, if people don't recognize it, people won't sort of understand the goals of this project. And so we're really, really working hard to make this known internationally. And this kind of recognition just supports that so strongly. So we thank NC3Rs and we thank GSK. And so thank you very much.
Thank you very much, Peter. That was an absolutely wonderful presentation, a really fantastic piece of work. And as you said, the potential for enormous 3Rs impact, which is what we're all about. But I think also potential for addressing many of the concerns we have about reproducibility of some data as well, and look forward to seeing this being taken up much more in the future. Now, I know we've got time for questions later, but we are going to move straight into Renata's piece of work and have her presentation before we open all of this up to questions. So just to say a few words of introduction to Renata. As you know, her piece of work was highly commended. So in other words, it was the runner-up, and it was a really splendid piece of work. And Renata comes from the Oswaldo Cruz Foundation in Brazil. Her paper is called a cytotoxicity assay as an alternative to the murine model for the potency testing of Bothrops jaraca venom and antivenom, an intra-laboratory pre-validation study. And it was published in March 2024 in Alternatives to Laboratory Animals. Now, this paper describes the methodology for a cell-based assay to evaluate the cytotoxicity of this Brazilian snake. And it looks at the reference venom to determine the inhibitory concentration of the antivenom needed. Now, alongside sharing this methodology for the assay, Renata performed a retrospective analysis comparing her in vitro data with existing in vivo data, demonstrating the reproducibility of the cell-based assay. The prize panel recognized the significant potential of the cell-based cytotoxicity assay to replace the current mouse lethality neutralization assay recommended by the WHO and the Brazilian Pharmacopoeia for testing the efficacy of antivenom therapies. So potentially a hugely important paper. So Renata, it's over to you. And I know we're having a recording of your presentation, but you are here in person, so really appreciate that. And of course, you'll be willing to answer questions afterwards. So if we could try and get Renata's presentation up, that would be great.
Good afternoon, everyone. First, I would like to thank NC 3R for the opportunity to be here to present our research and demonstrate how we can contribute to the search for approaches that do not involve animals. In addition, I would like to express my satisfaction and gratitude for the excellent news that my article was highly recommended for the prestigious International 3Rs Award. It's an honor to see that our efforts are being evaluated, serving as a great incentive to continue the search for alternative methods to the use of animals. This award, organized by NC3R, increasingly drives research on alternative methods that benefit science and society. Our work is aligned with the principle of the 3Rs and driven by the search for approaches that do not involve animals, aiming to mitigate the impact of animals' experimentation that involves death, pain, and suffering. This project is part of a validation study of a methodology developed at National Institute of Quality Control of the Oswaldo Cruz Foundation in Brazil. Here, we have a group dedicated to the development and implementation of alternative methods for decades. This study is part of my doctorate, which I completed at Fluminense Federal University at the Institute of Biology within the postgraduate program in Science and Biotechnology at UFF. Before talking about the article itself, I will contextualize the project. Snake bite envenomation is a serious public health problem and has been classified as category on the list of neglected tropical diseases. However, it's responsible for approximately 2,700,000 envenomings, around 1,038 deaths, and 4,000 cases of morbidity annually in several regions of the world. For more than 100 years, antivenoms have been the only treatment available for victims of these accidents, and quality control is essential to ensure efficacy and safety. To perform quality control in the antivenoms production process, as well as for the release of final batches, the use of animals for experimentation is a conventional approach and recognized as the gold standard. At least 200 million animals a year are used in scientific experiments. Brazil ranks 70th in this scenario, using about 2 million animals a year for research purposes. The antivenom production chain depends on the use of animals in several stages. Snakes are subjected to a venom extraction process. Rodents are bred to feed snakes in captivity. Horses are immunized to produce antibodies, and mice are used in vivo testing to determine the mean lethal dose and the mean effective dose. In the regulatory context, tests to determine the potency of antivenoms in vivo demonstrate inherent variation related not only to reliable and predictive power, which are questionable due to interspecific differences, and in relation to accidents in humans. Maintaining an animal research facility and experimental vivarium is costly. In addition to the long period between breeding and animal testing, the number of animals used is still very high. Although these animals play a crucial role in the production of antivenoms, the World Health Organization recommends the implementation of 3Rs with the following actions: reduce the number of animals used, improving procedure to minimize suffering, and replace them with alternative methods. This work directly responds to the host call for the total replacement of animal testing. The principle of the technique is based on the property of botrops venom to cause a cytotoxic effect on viral cells growing monolayers and antivenom being able to inhibit this toxic effect. The methodology adopted to quantify the cytotoxic effect of botrops venom and its inhibition by botrops antivenom in vitro is a modification of the cell quantification technique described by Marges and Borojevic in 1989. This method is capable of quantifying adherent cells on flat surfaces based on the adoption, elution, and subsequent determination of the optical density of the Comas brilliant blue dye that stains cellular proteins and lipids. The concentration of adherent cells can be assessed by evaluation of the dye, with the optical density of the dilute being directly proportional to the concentration of live cells adhered in a monolayer. It's possible to determine the amount of dead cells by comparing the OD of each dilution of the venom with the OD of the cell control. Through the dilute, we can observe the cytotoxic effect of the venom with greater cell death and the inhibition of cytotoxicity by the antivenom proportional to the greater number of living cells and consequently greater coloration. It was also demonstrated that the proposed method is capable of quantifying the cytotoxic effect of metalloproteinases and phospholipases A2 present in the venom of Botrops jararaca. These classes of toxins are known to be responsible for the main toxic effects of Botrops venom associated with both clinical accidents in humans and lethality in mice. All the experiments were performed in triplicates, and the results analyzed by using probit analysis. The OD values obtained were evaluated with the CombStats computer program. CombStats is routinely used by antivenom producers and quality control laboratories. The results were also evaluated for individual measurement biological assays with the SPC Explorer program. These control charts compare the replicate average of tests carried out in 4 different days and aim to show the variation between these consecutive groups over time. The values obtained were within the acceptable limits and remained closer to the principal line, which represents the overall mean, and the 4PL model showed a good linear response. The reproducibility of the results obtained with the current in vitro methods, as well as the need to replace the use in animals according to the 3Rs principles, make these in vitro methods a strong potential candidate to replace the in vivo methods. These results justify a future evaluation of interlaboratory reproducibility. The main impact to date has occurred in our laboratory, where we have successfully developed and pre-validated the in vitro assay. We have established the methodology, optimized the parameters, and demonstrate its potential as a replacement method through multiple independent assays. Our proposal is to coordinate an interlaboratory study that includes training professionals working in antivenom production to test the reproducibility of the alternative methods in their respective laboratories. A cost assessment was carried out for both methodologies, where the in vitro methodology proved to be up to 69% more economical, since the unit cost of determining the potency of Botrops antivenom by the in vivo method was estimated at $519. while determining the potency by the in vitro method was estimated at $182. When it comes to animal lives, they are priceless. The most important thing to consider is that replacement will save thousands of lives because approximately 2,000 mice are used annually only for batch release for botulinum antivenoms. Validation of a method consists of working with a quality management program with calibrated equipment, documentation, traceability, and reference standards. This project was carried out with great dedication and a small budget. It's a simple, cheap, and accessible method, and it was successful in pre-validation. My dream is to be able to conduct an international study with in addition to strengthening the validation of the method, would create a space for collaboration in several other related projects. Now, I wish to express my sincere appreciation to my collaborators for their exceptional contributions and expert guidance through the course of their research. Furthermore, we extend our thanks to the postgraduate program in Science and Biotechnology at Fluminense Federal University and the National Institute for Quality Control in Health at Fiocruz for their foundational support. Thank you for the award, and I leave here my contact, and I'm available for questions.
Thank you very much indeed, Renata. That was a super presentation and such an important piece of work. I mean, I think we all know what a problem snake bites are, particularly in your part of the world, but not just in your part of the world, as your map showed very, very beautifully. And we really look forward to seeing that system of yours being taken forward. So really all I want to say, just to bring it to an end, is firstly, huge congratulations to Peter and his team and to Renata and her team for 2 fantastic presentations and such incredibly important work with both papers with the potential to change the way in which we're doing things. And Peter, of course, has demonstrated that he's already well on the way to getting that change that I'm sure we would all like to see. But Renata, we hope very much that you'll be able to take your work forward and get the impact that I think we all feel that you truly deserve, because it's such an important step in the way in which we've determined the potency of those antivenoms, which we all know are very, very important. So a big thank you to both of you and your teams for all the amazing work you've done, and congratulations again. We look forward to hearing more coming from your labs in due course. Big thank you again too to Andy and GSK for their very generous sponsorship of this very important prize. And finally, of course, a huge thanks to the team in the NC3R's office led by Joanne. And none of this would have happened without them. They've been absolutely stellar right from the very outset. I wish we could do a proper clapometer on here, but they really deserve a huge round of applause for all the hard work they've done in making this run like clockwork from beginning to end, and really getting to understand the papers, the science behind them, and being able to contribute to the discussion in the way that Joanne led it just now. It's been a really, really great experience. So a big thank you, and keep going. And to colleagues who have been watching, we hope very much that next year we will see applications from you, and we can keep this prize going for many years to come. So thank you all very much indeed, and look forward to seeing you again next year.